Welcome. It's time to kick off. Thank you for coming to my little session. I've got about 40 minutes, and I hope to make genetics intelligible to everyone. It's a difficult subject for most people to get their head around because it's pretty esoteric, and most people didn't have much of an education in it. Most of the neurologists I work with don't have much of an education in it either. So, so don't be intimidated by it. The reasons for studying it and for... Please come on in here.
Sorry.
That's okay. The reasons genetics are important aren't probably the reasons that you're thinking about. You might be thinking about personalized medicine. You might be thinking about, why me? Why did I get Parkinson's disease? You might be thinking about prognosis. What will my life look like in 10 years' time if I know about my genetic predispositions to this disease? Right now, we really can't tell you many of the answers to those questions. What's important about genetics of Parkinson's disease is that it paves the foundation for the future in terms of developing therapeutics, in terms of developing medications that will slow or prevent the disease from getting any worse, or prevent it from occurring in the first place. So my passion for Parkinson's disease, I always say, is to predict and prevent. I've been working in this field. It's been very humbling, and in the genetics of Parkinson's disease now for more than 25 years. So in the beginning, to give you some insight, I'm going to take you through a little story of a particular trip that I've had, a journey, one of my journeys in Parkinson's disease. In the beginning, people said it was a waste of time. There is no heritability in Parkinson's disease. It's very low. You don't see Parkinson's disease really very often in families. But nevertheless, everything in biology has a genetic basis. Any biological organism has a genetic blueprint of its life, and it's everything else in I don't know. It's one form of truth, if you like. It's bedrock, and upon that, those particular discoveries, you can basically identify targets for disease intervention and for diagnosis. So I'm going to take you on a little journey, a little story, and tell you a bit about it. On the slide is a family that I was introduced to in 1997. I was working at Mayo Foundation at the time, and I went to see Manfred Munter. He was a neurologist. He was the chair of neurology in Mayo Scottsdale. And Manfred, and I was a newbie. I was a fellow, green as grass, really, thinking about what am I going to work on for the rest of my career. My background is in the genetics of complex traits. So what I do is study the genetic etiology of people who seem to have no genetic family history. I actually worked on cognitive dysfunction in the context of trisomy 21, context of Down syndrome, for my thesis, which is very different. I worked on statistical genetics. So when I came to Mayo, I was looking around and thinking to myself, I came there to work on Alzheimer's disease, and I realized that the field was very crowded. There was a lot of very senior people there who were very eminent in working on Alzheimer's disease, and there really wasn't room for me. So anyway, I went out to Mayo Scottsdale, and I met Manfred, and he introduced me to a paper that he'd been working on for the last 15, 20 years, on a family that he'd been working on for like 20, 30 years of his life. It's named after him. It was named the Spellman-Munter kindred. He inherited it, in a sense, from his predecessor. And he was writing up a paper, a biochemical paper at the time, on 4 brains, 4 autopsies that had come from this particular family. No one had figured out what was wrong. This patient, and you might notice that she has a masked face. She's holding the baby in a kind of strange way. She's got a little bit of kind of dystonic posturing in one arm. She came to Mayo in 1924. She has young onset Parkinson's disease, and almost every gene in Parkinson's disease that matters, from a clinical trial perspective, was identified in patients with young onset Parkinson's disease. Most of them, we know now, are with a family history of the condition. Although it may not be obvious, it may not be immediately apparent. Gene mutations have to be what we call penetrant. They have to manifest. And even gene mutations with the penetrance of, it has to be greater than 50%, maybe greater than 70%, to be able to see it manifest in a kind of Mendelian sense. So this may make no sense. You might remember Mendel and his peas. I don't know. There's basic genetics. Mendel came up with the idea that genetic traits can be passed down from one generation to another. There's dominant traits and recessive traits, and you can see these patterns in some families. But that's only for really, really penetrant causes of disease, and that's incredibly rare in most disorders, let alone Parkinson's disease. It's incredibly rare to find families with PD. So first question you might have, if I have a genetic risk factor for Parkinson's disease, what's the likelihood I'm going to pass that down to my kids and they're going to manifest the disease? That's what a lot of people are concerned about, worried about. The likelihood of that is very, very low. Very low. I wouldn't be concerned about it. Nevertheless, it's really important. This, to take you back to this story. So Manfred told me about this family. They come from a small town in Kingsley, Iowa. What they did at the time, and they were a farming family. But they were told by Mayo since 1924 onwards that basically that Parkinson's disease is an environmental problem. There's something on the farm. It's a bug. And you have to outrun the bug. And this was kind of like something that the family really picked up on and realized, because each generation of the family kept getting this early onset form of Parkinson's disease. The mean age of onset in the family is 33 years of age. And the duration of disease to death is about a decade. It's short and quick, and it comes on early. And it's at a time of life, of course, when people themselves generally have families and maybe have kids and themselves, and they need to work, and it's devastating, right? But the family listened to this, and they thought it was something in the well water, because at the time, and still, well water is thought, drinking from a well is thought to be a risk factor for Parkinson's disease, and so they tried to outrun the bug, and they dispersed. They left Kingsley Island, and they left the farm, and they ran off to all different parts of the United States. And when I started working at Mayo, Manfred introduced me to one or two members of the family that were still in contact and still getting treatment at Mayo. And there was a particular woman who was getting treatment at Mayo Scots who lived in Phoenix. She was a bank teller who just got diagnosed with young onset Parkinson's disease. And I also heard about some brothers that were living in Seattle. So I decided to go and visit them. And I went with a resident neurologist at the time, Katrina Gwynn, and took a flight. The first flight actually was to Seattle to meet the brothers. And we arranged with their mum that we were going to come and visit. In Seattle, if you know, getting there was quite a challenge from Florida. It's a long way, right? And we arrived late. It was getting dark. Got the hire car, driving out to this house. Got to this very run-down neighbourhood. Pouring down with rain, of course, because that's what Seattle is. It's always raining in Seattle. And knocked on the door, and I could hear this wailing, crying inside the house. And there's some commotion going on, and it was a nerve-wracking experience. And the two guys who answered the door, big burly guys, lots of tattoos. In Mayo, the term for it is disinhibition, because and it was a strange thing to ask them. They said, "What do you come for? What do you want?" And I said, "Well, I've come for your blood." and I don't think it was the right thing to say, because they got me in the back of the house, out in the back, and to have a few words with me, and to find out what my intentions were. And it was my intentions, of course, were to try and map the disease gene that I thought caused the condition in their family. Right? Because I could see from the picture of the family, and I'll show you it, this is what we worked out, that it was a Mendelian dominant pattern of inheritance. like Mendel's and his peas. This was the lady you just saw in that photograph that I pointed out with young onset Parkinson's disease. She's now the grandmother in this particular pedigree. It's her husband, her children, and then obviously the grandchildren. And anyone with a little black symbol here has Parkinson's disease. And half of each generation has Parkinson's disease going back. And it affects men as well as women equally well. And the ages of onset, you can see here, 32, 46, 24, 31, relatively young, right? And so when I was out there in the Florida room kind of thing, the back of the house, these guys told me their history, and they told me about how they'd been poked and prodded. They'd had Mayo physicians come and visit them over the years, teams of nurses that had all this testing done, and they'd never come back. They never had a satisfactory explanation of what was going on. They never had a diagnosis for this or a reason why it was their family. They were told it was environmental. And I said, we can do better. But I do need that blood. We can do better. We can figure it out. And we can come back and we can develop, I think, a therapeutic based on that, because we can identify a target. If the family's big enough, if we can map the gene. Now this is like in the late 90s. Gene mapping then was much more challenging than it is today. Today I can sequence 20 of your genomes, actually 40 of them, in a single day in my lab. It's easy because the technology's changed enormously, you know. Back then you couldn't do genome sequencing in individuals like that. It wasn't nearly as expeditious. Anyway, kind of long story short, we went to visit basically every branch of this family. This branch of the family came through, there was a lot of investigative detective work going on. This branch of the family came through a pathologist, a neuropathologist, Millers and Water. What was it? Waters and Miller. Sorry, Waters and Miller was the name of it. It's a neuropathologist and a neurologist living in California, and they described the brain that they'd basically been studying with really fulminant Lewy body disease, lots of Lewy body pathology, which I'll show you in a minute. And it turned out that the brain was from this lady, and we had one of the nursing assistants actually managed to figure out the genealogy, that they were linked all the way back up here to part of the same branch of the same family. Pretty impressive. It took me 6 years to map the gene in this pedigree, which is a lot of time and a lot of effort. And part of the reason was because we got it wrong in the beginning. We didn't have enough family members taking part. And I'm not talking about affected family members, I'm talking about unaffected family members. We couldn't figure out that different branches of the family, how they were related to each other, because we couldn't see the flow of genetic information through the pedigree. There was too much missing data. And we solved that ultimately, but it did take a long time. And I went round and I visited every branch of the family. And it was humbling, really humbling to hear their story about their disease. And it had a huge impression on me to do something about it. All right.
What's the difference between the solid squares and the half squares?
Oh, okay. What's the... Okay, they're all quarters. Yeah, they're all quarters. So this is Parkinson's disease. The black symbol, that's all that represents. Okay. And the squares are males, and the circles are females, and the diamonds have just disguise the gender. So you don't know if it's male or female. The oldest person in the SIB ship is generally on the left-hand side, just give you a bit more information, more than you want. And then the youngest down this side, and that's just standard. This is drawn wrong. I always put the males on the left because the women are always right. My mother taught me that, and my wife has been feeding it into me. So it's pieces of the puzzle. That's what I'm talking to you about. We mapped the gene, and in 2003 we published it in Science, and this is not a picture of the paper, but it's kind of a picture from the lab. And seeing is believing, right? And it turned out that the gene itself was alpha-synuclein. It was an alpha-synuclein multiplication. It's a little gene on chromosome 4q21, it's a chromosome position. And these family members, unlike you and I, well, I think, the chances are that all of us in this room have 2 copies of this gene. We have 2 copies of every gene, pretty much, right? We're diploid. We have one set of genes from mum and one set of genes from dad, and they make us. So we've got 2 sets of everything. We've got 2 sets of alpha-synuclein. And this here, what is shown here on the left-hand side of the slide, are in blue is the nuclear DNA material, actually in white blood cells. So this is just some some leukocytes, some white blood cells, basically on a microscope slide, stained with the dye, DAPI, which basically gives it this blue coloration if you illuminate with the right light. And what I've done is probed it for the alpha-synuclein gene, and I've got 2 probes, one green, FITC, and one red, rhodamine, and they're for 2 fragments of the alpha-synuclein gene. And where they coalesce and line up together, they give you a bit of a yellow signal. It's either green, red, or yellow. So you can see here, this is a normal individual. They've got 2 copies of the alpha-synuclein gene. This is an individual who's actually got 3 copies of the alpha-synuclein gene. You can actually count them, right, in an interphase nucleus on a slide. And this is a member of the Spelman-Munter kindred, and they have 4 copies. 1, 2, 3, 4. 4 copies of the alpha-synuclein gene. That's the only difference between them. Well, not the only difference, but that's the reason they have Parkinson's disease. They have twice as much, 100% more alpha-synuclein. Right? And that's why they get the disease young. It turns out, by studying many, many, many more people who've taken part in genetic testing over the years, that there's a beautiful relation. Well, I don't know if it's beautiful, but there's an what's the right word for it? There's a really robust relationship. Thank you. Scientists get a bit carried away sometimes. There's a really robust relationship between the numbers of the copy number of alpha-synuclein and the age of onset and of motor phenotype, of motor symptoms of Parkinson's disease. So if you have 3 copies of alpha-synuclein, you're probably going to get the disease typically 65, 70 years of age, when most people get Parkinson's disease. It looks like garden variety Parkinson's disease. If you have 4 copies of alpha-synuclein, you get the disease younger. You get it here. You get it in your 30s for the most part. Maybe as late as 40, and 48 I think is the latest age of onset we've had for somebody who's got 4 copies of the alpha-synuclein gene. So it's predictive. We can genotype an individual before they even manifest symptoms, and we can say, you're going to get it, and we can say approximately within 5 years or so, say when you're going to get it. And we can also say something about the evolution of the disease, because in these family members with synuclein multiplications, there tends to be more cognitive problems, more dementia, and it comes on a little earlier. But it's not a clear-cut relationship. So there's some individuals out there with alpha-synuclein multiplications who presented with what we call dementia with Lewy bodies when they first came in. They presented with cognitive problems, and Parkinsonism really wasn't the issue. It was the dementia was more of an issue. But there are other patients that have come in who've had the disease now 20 plus years, and they've still got a motor phenotype. They don't have any dementia. It's amazing. There's no relationship like there is with motor onset, with the MCI, with the mild cognitive impairment, or with the dementia onset. So what about, this is one family, right? So what? Who cares? That's what people said to me. Editors of journals, reviewers of my grants. Who cares? It's not important. It's just going to be one family. It's so rare. What does it mean to garden variety Parkinson's disease? Most people with Parkinson's disease. What is it about that and typical Parkinson's disease? And it's this. This is the only gene that's ever been found to date. Right? Yeah. I think that is not just associated with age of onset of the motor features, and there's a beautiful relationship with that, with dose and age of onset, but it's also associated with the pathology that you find in the brain of individuals with Parkinson's disease when they die. So when Parkinson's disease in part is a pathologic diagnosis, and if a neuropathologist looks at the brain of people with Parkinson's disease, 90% of the time they find Lewy body pathology in the midbrain, in the brain stem of those individuals. They see the loss of the black substance, as a substantia nigra, and they also see these inclusions. This is a typical Lewy body, and this is you can see a little bit of melanin here. This is actually a nigual neuron, and it's a cell body of a nigual neuron. It's in the midbrain, and this is what a Lewy body looks like. Some people call them Levy bodies, and my pronunciation's maybe wrong, but it's got this central dense core, and it's got this radiating halo, if you like, of filaments. And the brown staining here is actually alpha-synuclein. So antibodies raised to alpha-synuclein when you throw them against a slice of tissue from the brain of somebody with garden variety Parkinson's disease, this brown stuff, it all lights up. Right? It shouldn't be there. None of this brown stuff should be there. This should be just blue-gray, the whole slide, right? I don't know if you can make out the cells. These are cell nuclei. And this is what you typically find in Parkinson's disease. These are cortical Lewy bodies in dementia with Lewy bodies. A different part of the brain. This is the temporal lobe. This is a higher magnification power view of actually a Lewy, what we call a Lewy neurite. So this material, this alpha-synuclein, because this brown substance is alpha-synuclein, basically fills some neurites, some axons, processes in the brain, and obviously it's not good. It's not good. But we don't know it's causal of the disease. We know it's causal of the disease in these patients with alpha-synuclein multiplications. But in garden variety Parkinson's disease, I'm not so sure. And I've been labeled a heretic. Sometimes I am heretical. I stand up and I told John Georgianowski and Virginia Lee last time that I think it's an epiphenomenon, right? And there's a reason for that. There's a good reason for that. The tombstone is not generally the cause of death, right? If you go to a graveyard, you see a lot of tombstones. It's not generally. This is a tombstone. This is telling me that these neurons are unhealthy, they're sick, but maybe the reason the synuclein is there is because this is the final kind of stages of those neurons trying to upregulate a particular protein, alpha-synuclein, and package it up to prevent them from dying, right? So when we first got this picture, this was in 1998. Actually, it was a fellow in Cambridge, Michel Goddard, and Maria Spellantini, a couple in Cambridge. They'd been working on the alpha-synuclein family. It's actually 3 synuclein genes, alpha, beta, gamma. And they've been developing antibodies for these. It was originally described in Alzheimer's disease as the non-amyloid component of plaques. And they made these antibodies, and we got some. And Dennis got some, and I remember him coming running. Dennis was the neuropathologist at Mayo. He came running downstairs, and Dennis is very, I don't know. If you see him at a Jags game, he's not, he's a different beast. But for the most part, he's very upstanding and very sober and very, But he came running downstairs. He was so excited. He'd never seen anything like this at all. It was incredible to see this pathology or what it is. Since then, we know from a variety of studies, and Heiko Brack's anatomy studies are probably the best described, that the alpha-synuclein pathology that you can see in the brains of individuals with Parkinson's disease seems to occur, we don't know for sure, but seems to occur in the body and may ascend up into through the brainstem and into the midbrain and then ultimately into the cortex as the disease progresses. And I don't know if you can see the slide, but this is Brack stages 1 and 2, and it's talking about autonomic and olfactory disturbances. So problems with constipation, maybe problems with loss of sense of smell, sensory loss, things like this. No motor diagnosis. And the synucleinopathy here is in the brain stem. It's in the olfactory bulb, right? That brown staining that I just showed you. This is stages 3 and 4, some sleep problems and some early motor disturbances. So maybe REM sleep behavior disorder, and very early, maybe not, suspect as Parkinson's disease for a diagnosis, but the synucleinopathy now in the brainstem, and it's getting up to the midbrain, and you're starting to see cell loss. And of course, in the late stage of the disease, many patients go on to get cognitive impairment and dementia, and it seems to correlate with the spread of synuclein in the brain, right? But it's not a hard and fast relationship. Like I said, there's some patients out there with synuclein multiplications who don't have dementia, and they've had the disease for 20 plus years since their motor onset. So the other piece that's important in terms of synuclein in the general population, it's probably the most robust in terms of statistical significance gene for Parkinson's disease that's ever been described. In the general population, the studies are huge. I mean, something on the order of 40,000 people have taken part, patients with Parkinson's disease, and more than 1 million control participants have taken part. And we can see there's differences in the frequency of variants within the alpha-synuclein gene. This is a study that we did in 2015 or so, where we resequenced 135,000 nucleotides worth of sequence around the synuclein gene, and we got every single genetic variant that's different. We all have genetic variability. That's why we look different, right? It affects everything in our life. We actually have about 5.5 million variants that are different between me and you, between all of us. And if you look at this piece of sequence of alpha-synuclein, you can find all these little variants. And then in this particular study, we tested them in the context of patients with Parkinson's disease, patients with mild cognitive impairment in Parkinson's disease with dementia, and healthy individuals, and look for differences in frequency of how often you see those particular variants occurring. And what we found is that there's variants at what we call the 3' end of this gene that were associated with Parkinson's disease, and there's variability at the 5' end of this gene that's associated with dementia. And that's just in the general population, right? But this as a risk factor in the general population only has a very, very, very tiny contribution to risk. Minute. It's not segregating down the family line. You don't see it in families, all right? It's not useful for any diagnostic test, right? But it's an interesting thing to know, because if we develop a therapeutic for alpha-synuclein to remove that pathology, for example, or to lower its expression down, chances are it's going to work in people with general garden variety Parkinson's disease too, not just that spermiotic kindred. That's why it's important. So can these insights stop Parkinson's disease? I wouldn't have spent 25 years of my life working on it if I didn't believe that it couldn't. I think it can. And this is just one gene, right? So you had a mention of this earlier in the clinical trials talk, which was very good in the first presentation. There's a number of companies out there right now with drugs in different phases, phase 1, phase 2, phase 3, whatever, for therapeutics to lower synuclein expression. It's actually being targeted, and there's 2 ways of doing it. You can lower the gene level, or you can lower the amount of protein that the gene is producing. So genes are transcribed into mRNA, and mRNA is made into protein. And you can target either of those things at any stage. And so these are just some of the trials. The battle begins. This is a paper from, I think, 2017 or 2018. And it's still ongoing, right? It's going to take a while. For Alzheimer's disease, it took more than a decade. And then we had some positive news from aducanumab and subsequent antibody therapies that it is having benefits in terms of slowing dementia progression to try these antibody-based therapies. I think we're going to see the same thing in synuclein. It's not going to be for synuclein and for Parkinson's disease. It's not going to be a fix, but it's going to slow progression of the disease down. All right? But what we need to really do is to understand what synuclein is doing in the body and in the brain, and what's its true function. And that's something I do on a daily basis. It's taken a while. So how do you fast track this precision medicine idea? You know, we need more targets. It's not enough to have just alpha-synuclein, because the bodies are very complicated. And we've got 20,000 genes. There's a reason we've got 20,000 of them, because they work together in concert. And one of the things I like to show people, this motor, because it's a movement disorder. Motors are responsible for movement, right? Of course, you all picked up on that before I even mentioned it. But and I've labelled the component parts, and the alternator and carburetor. I can't even see the carburetor, but anyway, the different component parts of a motor car, of a motor engine, that make it work. We've got to figure out what that is, all right? And I've just labelled up here alpha-synuclein, BPS35, leucine-rich repeat kinase 2. These are major genes for Parkinson's disease, in which genetic variability if you have it, can predict your diagnosis or can secure your diagnosis. People will say, the genetic counselor would say to you, okay, yeah, that's the reason you have Parkinson's disease. And can also predict maybe Parkinson's disease or its occurrence, its likelihood of its occurrence anyway, in other relatives of your family. These are major genetic forms of Parkinsonism. There's about 40 of them now. It's quite a lot. But there's a lot of components to an engine, right? And until we figure out what the parts are in that engine, we can't figure out how to put it together. It's really challenging, right? How does it work? Now, I'm a great believer, and I might be wrong, that all roads lead to Rome. It's an old saying, right? And that all of these genes basically point to the same common problem in Parkinson's disease, because they manifest, ultimately, everybody with PD is different. Their diagnosis is different. Their presentation is different. They have different non-motor and motor components of the disease at different stages, right? But at the end of the day, we can label it and we can say, oh, that's PD, that's PD, you got PD, you got PD. We can see it, right? So I think these genes basically point to the same pathway that's affected in Parkinson's disease, and it's becoming clearer and clearer what that is, but we don't have it yet. And what we need is people with Parkinson's disease and their family members, and especially people with younger onset Parkinson's disease. They may not be young now, but to take part in these genetic studies, because without them taking part, we won't have these new therapies, and we need them. We need them. It's really important. So yeah, out of the roadmap. So once we have a gene, as I mentioned to the Spelman-Minter kindred, we want to do something about it. Okay, it's useful as a tool for diagnosis, but what do you do with it next? So one of the things we do with it next is we basically put it into cell models. I do a lot of work on genetically modified mice, and I try and make the mice as etiologically and as physiologically faithful to the human condition as I possibly can. They don't get Parkinson's disease. I don't expect an animal who lives 2 years long to get Parkinson's disease. It's not going to happen, right? Or a mouse. Mice don't get Parkinson's disease. But what they are is models of gene function and gene dysfunction. And they can tell us about the biology of what that particular protein is doing in the brain of those organisms, and perhaps in their gut as well, and in their immune system. So we make these genetic models. And we're basically doing a lot of studies on these mice to try and figure out how their brains compensate, in part. This is a study here of a rat's brain. And what you see here in these little black dots is the striatum of a rat lit up with 18-fluorodopa PET imaging, positron emission tomography. The rat has the LRRK2 mutation. So we're measuring LRRK2 biology. Different gene for Parkinson's disease, but probably one you've heard of. Now I thought, that's my presentation, but I thought I could tell you some other stories about, if we've got time, I don't know what time it is, I can talk for hours. 12 o'clock. How are we
Can I
doing?
ask a question?
Yes, sir.
In the gene map of synuclein that you do, are any of those sequences removed when the gene is expressed? Are they spliced out or anything like that? Is that
So there is alternative splice. See what I mean? There's always going to be somebody, isn't there? Yeah. So our alpha-synuclein, the nomenclature's changed a little bit. Has 5 exons. The way genes are arranged, they're not singular units. They have what we call exons and introns, and there's 5 exons. An exon 3 and exon 5 are alternatively spliced, and you do see an exon 3 deleted form, an exon 5 deleted form, and an exon 3 plus 5 deleted form. Those isoforms are far less abundant. They're pretty rare, but I think they're interesting, especially the exon 5 deleted version. The exon 5 version, alpha-synuclein is to tell you something about its function. It basically has what we call it forms an amphipathic helix at the 5' end, the N-terminus, and that binds to lipid. And what it does, it helps to keep vesicles, little balls, if you like, balloons of material surrounded by lipid It helps to keep those vesicles separate from each other, and it helps regulate exocytosis, the release of those vesicles from the brain from synapses. And your question about the exon skipping and different forms is important because the 5' end, so the 3' end of synuclein, the C-terminus of synuclein, is an acidic tail. The 5' end is its amphipathic helix, the membrane. The acidic tail is really important. If you remove the acidic tail, you get a form of synuclein that has a much higher propensity to aggregate. And Maria Spielentini actually showed this probably best. She made a mouse model. She removed the last 20 or so amino acids of alpha-synuclein. She put that into a mouse and masses of synuclein pathology. Right? It looked like MSA rather. It was not just neuronal pathology, it was oligodendroglial pathology. It was, that 5' end is, this C-terminus is very important. Yeah. So skipping might be important. We don't really know. You're going to stop me, aren't you?
I'm just saying, you have please wrap up.
Please wrap up. Please, question.
Can I ask one question? Yes. So I participated in the genetic study that was done through Fixel.
Right.
And they said, well, maybe in 2 years, we would get some results. Is that the case? I mean,
have you asked about those results?
No.
To Fixel?
No.
I recommend that you do. I knock on the.
I guess I assumed that they would tell you.
Okay. So for the most part, that's probably it's going to come to me, isn't it? But for the most part, most individuals who take part in these studies, they're ordinary. They're nothing to be concerned about or worry about. We don't find anything. It's very important for the totality of the study, but from an individual point of view, there's nothing to tell you. If we call you up, I'll send you a letter and say we've got some news, but would you really want to know? That's when you might want to be more concerned. So no news is actually good news in this case, right? We have been looking at patients with young onset Parkinson's disease specifically. We find that between 1 in 5 and 2 in 5 of those patients actually have a genetic form of Parkinson's disease. It's very high. I think it's very high. But people with later onset Parkinson's disease, it's very rare to see monogenic single gene forms. So one of the things, some guidelines for you in genetic testing or getting involved in genetic studies, you have a right, it's your DNA, you have a right to your information, right?
Yeah.
And if you want it, you should ask for it and have it. The challenge is it's a whole genome is 150 gigabytes of data. What are you going to do with it? I mean, I have a challenge of what am I going to do with it? It's a lot of data. It's like stars in the night sky. So one of the things that's important to do is to make sure if you're involved in a genetic study, find out where that information is going. It needs to be shared. I'm not one of these people who's private about my genetic heritage or background, and people think about this differently, right? But I would want that genome information to be shared with as many investigators who are willing to look at it and make sense of it, because it is like looking at the night sky. Somebody can focus on, Saturn, and somebody can focus on, Betelgeuse, you know what I mean? And it's going to take a while to figure it out. Maybe a little, machine learning magic, but it's going to take a lot of expertise from a lot of people. It's also going to take a global effort. So I was going to tell you about some of my stories about, I just told you about one family, but we've been involved in mapping many of these genes. And each of it's been such an amazing experience to me to go to people's houses and visit them, and their generosity in giving a blood sample and some clinical information. And it's led to these discoveries. It's huge. It's huge. So.
Yeah, I have a question that came up in my family just recently when I told them I had Parkinson's. They asked the question, is there a direct relationship from a DNA perspective from one generation to another? You covered quite a bit of information, but kind of in your own thought process, and let me give you an example. A grandmother shook like a leaf, somebody in between shook like a leaf. She was 64 when she passed away.
Right. So
I haven't been able to discover anybody else that may have had Parkinson's, except me.
So it does skip generations. It isn't necessarily penetrant. The most common genetic risk factor for Parkinson's disease that it doesn't. So you have to think about genetic risk in different ways, right? It's not all the same. Some gene mutations have really major effects, right? And they're causal, and you can see them segregating down the family line. Most genetic variants don't have major effect. It's very, very modest. And even cumulatively, in the entire genetic background, you really can't see what the effects are. I liken it to the family face, right? You look at your face and you look at your kids or your brothers and sisters and your parents, you can see there's a genetic relationship. Well, most of the time, right? You can see there's a genetic relationship. The family face. Everything else perishes, but that lives on in the next generation. The next generation, they have your nose, or they have your, Megamind forehead, or whatever it is, and it lives on. But that's not a monogenic trait. That's not caused by a single gene. That's caused by many, 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 many very modest variants affecting how your face looks. There are single genes, though, and single mutations that might give you a really big nose, or a unibrow, or one tooth in the front. Absolutely, absolutely. But they're very, very, very rare. And it's the two things together, if you like, this background of genetic variability and then major monogenic single gene effects together that make us who we are, not just for Parkinson's disease, but for any biological trait, right? So you might not see it in, you'll come back to your question. It's the penetrance of a particular gene mutation is it's very difficult to predict. Even if you had a gene mutation diagnosed, for example, if you had a LRRK2 mutation, it wouldn't necessarily say that you'd get Parkinson's disease. So Sergey Brin has made this public. He's the CEO of Google, probably one of the world's richest men. He's probably got $100 billion, right? He has a LRRK2 G2019S mutation. And he's terrified that he's going to get Parkinson's disease. His mum has Parkinson's disease. His aunt has Parkinson's disease. And he's spending $100 million a year funding work in Parkinson's disease. A lot of the genetic push is coming from him because he's funding it. He's funding it. He asked me to work for him 10 years ago, and I turned him down. Big mistake. Career suicide. But never mind. But it's because he's got. But there's no guarantee that he's going to get Parkinson's disease. Many. Only 25% of people with a LRRK2 G2019S mutation, by the time they get to 80 years of age, ever manifest symptoms. 75% of them won't, right?